Hi everyone. Today we're going to review cell-mediated specific immunity, also known as cellular immunity, and we're going to review how it relates to T cells. In this process, we're going to also cover the relevant parts of your textbook that go over cellular immunity in both bio honors or anatomy. In this lesson, we're going to address how cellular immunity is critical to your immune system and its regulation, and we'll cover cellular immunity and T cells related to it, and then we'll put cellular immunity and humoral immunity in context with each other to what we've learned about B cells and T cells, and then we'll talk a little bit about how COVID is involved in all of this. So just to remind you, specific immunity is the third line of immunity after a bacteria or a virus or any other pathogen has gotten past your innate defenses and somehow started invading your body and infecting your tissues and cells. And then when a pathogen has gotten past and is actively infecting your body and reproducing and stuff like that, we have two kinds of immunity involved in specific immunity, and that's humoral immunity and cellular immunity. We've talked about humoral immunity, which is when B cells and the antibodies they create basically attack a virus or bacteria or any other pathogen that's outside the cell. So oftentimes when it comes to viruses, at least, it's about finding a virus before it has entered a cell and preventing it from entering a cell. And cellular immunity is what we need when a pathogen has entered the cell or if your cell has become abnormal. In this case, because humoral immunity, your antibodies can't help against stuff happening inside the cell, we need a separate branch of your immune system, your T cells, to look inside your cells and destroy any cells that are abnormal or infected. So, if attackers get past your B cells and the antibodies they produce in your blood and they're inside your cells, you're going to need those trained assassins, those T cells, specifically killer T cells, also known as cytotoxic T cells, to destroy those infected cells. So, cellular immunity is all about T cells. They're the immune response you need against infected cells by pathogens. They're also the immune response you're going to need against any cells that are non-self, that are not the way your normal cells should be. So cancer cells and, unfortunately, cells that have been transplanted from transplanted organs will be attacked by your T cells, this cell-mediated response. And it's going to be different than, we actually have a cell response in our non-specific defenses, because if you remember, your non-specific defenses in the second line of defense also use a cell-mediated response through your white blood cells called macrophages, neutrophils, and basophils, non-specific white blood cells. So when we talk about cellular immunity, we're mainly discussing specific cellular mediated immune response from your T cells. Specific meaning the T cells that are specially made to only attack a certain antigen, a certain pathogen. All right, so those T lymphocytes, because remember T cells are kind of lymphocyte, we have a few major types. We have naive T cells, which are the baby T cells that aren't doing anything yet. All right, but when you activate them through an infection, so when you're infected, your naive T cells get engaged and they become what's called effector cells. Effector cells are going to do the active fighting against your virus, against your bacteria, against any pathogen or problem. All right, so we have helper T cells. Helper T cells are T cells that are involved in alerting your immune system, and then they're also going to be really huge in activating your immune system. So your B cells and cytotoxic T cells are actually engaged, promoted, and activated by helper cells. Then we have the big players, our assassins, the cytotoxic T cells, also known as killer T cells, and those directly attack or kill infected body cells. You don't really need to know T4 cells or T8 cells, but sometimes when you see images, we just a shorthand called helper T cells T8 cells or T4 cells or CD4 cells, named after a special cellular tag on them. And sometimes cytotoxic T cells are also nicknamed T8 cells or CD8 cells in pictures. So just a heads up, if you see those on images, that's what they're referring to. But in our class, you only have to worry about helper T cells as the name and cytotoxic or killer T cells as the other name. And finally, the last major class of T lymphocytes is what's known as memory T cells, which are the T cells that retain immunity for faster, stronger response to a pathogen or antigen if that pathogen or antigen returns, just like memory B cells, right? So these initial effector cells, just like the effector cells of the B cells, the plasma cells, they both take about a week to 2 weeks to engage against an invading pathogen. But once they have gotten rid of that pathogen, you have lingering memory cells that are going to stick around. And if they see that antigen, that virus, bacteria, whatever, they don't need 1 to 2 weeks to engage. They rapidly respond immediately against that pathogen to smack it down. And that's what we consider immunity, the presence of those memory B cells and T cells in your body. That's going to be what prevents a virus from reinfecting you, or if it does reinfect you, make it not as bad. That's what we consider immunity. So, helper T cells are critical to activating both humoral and cellular immunity. So, T cells are often considered part of cellular immunity. However, just a heads up, when your T cells are activated, when they detect an infected cell or they detect a active infection because your white blood cells, your phagocytes, have somehow eaten a pathogen and are showing that pathogen off, your helper T cells get activated, and then they send out signals. And just reminding you, the signals usually used to signal to another white blood cell to turn into something, those are cytokines. Cytokines are special protein signals that help engage your immune system. Anyways, those helper T cells then release cytokines to also activate other parts of your immune system. In this case, they'll help make the stimulate cytotoxic T cells to develop, and they'll stimulate fellow B cells that attack the same material. Okay, so I mentioned that earlier, but this is just to put it all in context. And generally speaking, a major sign of infection is elevated T cell counts, because you have a lot of T cell in your blood. If you're trying to attack something, you're going to make a ton of T cells to attack an active virus that's in your body or active bacteria in your body. And you'll also see high concentrations of cytokines because all of those white blood cells are signaling to each other to attack. All right, so how do T cells know if a cell is infected? It's all thanks to this protein that all cells are required to have in your body, and they're called major histocompatibility complex proteins. These are proteins that are found on the surface of all your cells, and they are actually identification badges to mark your cells as yourself. Because remember, a big part of your immune system is telling self from non-self, and the way they do it is by making all your cells have an ID badge. So the MHC works as a unique ID badge, unique to individuals. So my MHC tags are different than your MHC tags. So if I were to somehow donate tissue from my body to you, that tissue I donate to you will have the wrong ID badge, the wrong MHC tags, and your body's immune system will destroy whatever I donate to you. So whoever has a tissue organ transplant, they actually have to take immunosuppressants to suppress their immune system, to suppress their T cells and B cells from attacking any transplanted like organs they may have. Another side effect of those ID badges is it turns out that they're not just ID badges. Those ID badges, those MHC proteins, are required to randomly show a snapshot of what's going on inside the cell. Those MHC proteins actually display random stuff from the cytoplasm, from the insides of the cells, onto the surface of the cell. And the T cells will check the MHC proteins for the correct ID, and it will also check the random stuff those MHC proteins are holding up to check if the proteins being made by your cell are normal. Okay? So the MHC protein is the ID badge, and this ID badge is required to show whatever's randomly happening inside the cell. If the stuff they're holding in the MHC protein is incorrect, is an antigen, is non-self, then your T cell will recognize it and activate and help destroy that weird messed up cell. Whether it's a cancer cell or an infected cell, it's going to destroy it because it's making stuff that it shouldn't. So there is an issue here. T cells can only detect antigens if they are held inside an MHC protein. So that's easy when it comes to a cancer cell or a virus infected cell, because cancer cells and virus infected cells are making proteins for that cancer or for that virus. So they're going to have messed up antigens. So any random protein you pop off on the surface of the cell will show and be able to be detected by the T cell. But what happens if a virus isn't in the cell and it's outside in your body fluid, or a bacteria isn't in your cell but outside in the body fluid? While you do have antibodies and B cells there, your T cells can still help out. Why? Because you have phagocytes, because you have white blood cells that are randomly eating stuff in your blood during the second line of defense, during your random white blood cells in the non-specific immunity section. You have what we call antigen presenting cells, cells that present antigens of pathogens they swallow on their own MHC proteins. So this antigen presenting cell could be a phagocyte, a macrophage, a neutrophil, a dendritic cell that has eaten a bacteria, swallowed it, and destroyed it. And then it will take the random parts of the bacteria or virus it destroyed and put it on its surface, and then it will activate that T cell. Remember, activating a T cell, especially a helper T cell, whether it's directly activated by an infected cell or an antigen presenting cell that has swallowed a virus or bacteria and broken it down, either way, once you activate that helper T cell, it's going to go and activate all the B cells and all the cytotoxic T cells that will help destroy that virus or bacteria. So getting it activated, whether it's by an infected cell or by a virus or bacteria that hasn't infected anything yet but has just been swallowed and destroyed, you're good. You got your T cell activated, and now your cellular immunity is a go. All right? So just a heads up, different types of antigen presenting cells. We have dendrites, we have macrophages, and all of them are going to help activate other T cells and B cells to an extent. And also, actually, your B cells also work as antigen presenting cells too. So they're another type of antigen presenting cell. Either way. All right, so just reminding you, you have your infected cells and antigen presenting cells like B cells and phagocytes that are going to help activate your T cells. And once your helper T cells especially are activated, everything else gets upregulated. Everything else gets to blow up and help attack the virus even more, or the bacteria even more, or whatever even more. All right, now when we talk about cytotoxic or killer T cells, their job is to destroy body cells, destroy cells that, and to an extent bacteria, but mainly your own body cells that have an antigen. And if it sees it, what it's going to do is it's going to bind, recognize that this virus infected cell is bad, and then it's going to basically send a bunch of protein chemicals at it to help encourage that cell to die. That's going to cause what we call lysis, or cell bursting. The actual main protein weapon that it uses is something called perforin. You don't have to know that, but just a heads up, there are proteins by the cytotoxic cell that is used to burst the cells of infected cells apart. So those cells can no longer manufacture dangerous viruses. But then those viruses, again, will be in your blood, but that's all right, because you have your humoral immune system and the B cells you have making antibodies to block and destroy the viruses that are released from inside the infected cell. So you stopped the factory from making the virus, and you stop the viruses once they're in your blood. All right, so just to remind you, your immune system response. So a pathogen gets into your body. That pathogen is in your blood, and then your humoral response will attack it. Or it may be infecting a cell, and then your cellular response will take it on. Or it may be eaten up by your innate immune system, your macrophages, your dendrocytes, or something like that. But then, when those are eaten up and swept up in your second line of defenses, that will also impact your helper T cells. And your helper T cells are going to go in and help make your cellular response and your humoral response extra good by activating the B cells and T cells that are critical to them. And those B cells for your humoral response are going to make the antibodies you need through their plasma cells, and they'll have some memory cells just in case you get reinfected in the future. And your T cells that are activated by the helper T cells, specifically the cytotoxic T cells, are going to help actively kill things. And again, you'll have some memory T cells for future infection in that game. And all of that is thanks to those helper cells within your immune response. So if you destroy the helper cell, you kind of ruin all the major arms of specific defense. Okay? And this is like a slightly nicer version of that last one if you want an image of that last diagram in your notes. That's kind of cleaner and has like more detail. So revisiting cellular and humoral immunity, where humoral immunity was about infection or antigens and pathogens found outside the cell, while cellular immunity was about stuff inside your cell being infected and removing that. So humoral immunity is all about B cells and the antibodies they produce. And those antibodies are going to allow neutralization of the pathogen, meaning binding the pathogen and preventing it from infecting or doing anything bad. It's also going to help result in lysis of the pathogen. So lysis not of the host cell, but the pathogen itself. This is usually bacteria cells. And you can use your complement proteins to pop them instead of the perforin proteins used by T cells. And you're also going to create phagocytosis, meaning those antibodies are clinging on to stuff outside your cells. It's going to help signal your white blood cells to eat it. Okay? With cell-mediated specific immunity, you have your T cells recognizing abnormal antigens on the surface of your cells that are either infected or cancerous. And it's going to then result in lysis, also by complement proteins, in this case, complement proteins like perforin. And it's going to help result in enhancement of cell-mediated immunity and humoral immunity overall to help prevent spread. So when you have a pathogen, your job is you want to make babies. So you're going to have to deal with the fact that the host you want to infect, if you were a virus or bacteria or whatever, has an immune system. So you need to evade it for as long as possible. So there are multiple strategies viruses have and bacteria have evolved over time. And the ones that have done the best job at destroying or evading the specific immune system are the ones that are really scary when it comes to getting infection. And sometimes those viruses are the ones we consider lifelong viruses. Once you get infected by them, you're always infected by them. So one of the strategies that almost all viruses have is they can prevent infected cells from using MHC proteins, or at least reducing the amount of MHC proteins that they produce. So it makes it harder for that infected cell to alert the immune system, to alert the T cells. So COVID-19, among many other viruses, actually do this. Not great, but it does do it, which is why they can infect for a little bit. But the really scary ones are the other two. So HIV does a really good job of basically destroying specific immunity from removing it because it actually directly attacks the lymphocytes, particularly T cells. And some cells, and this is the other one, not cells, some pathogens also create permanent infection by actually sneaking their viral genome into the host DNA, but don't make new viruses. And by sneaking into the host DNA, they become what we call sleeper agents, lying in wait, because your cells can only detect infected cells if they are making weird stuff that'll be displayed on the MHC proteins. If they don't make weird stuff, then they won't be caught by the T cells checking the MHC proteins, and they become sleeper agents lying in wait, and they don't get attacked until they start producing viruses. And then if they do start producing viruses, they'll produce enough before the immune system gets engaged, and they'll just randomly pop up and have random cells start making viruses and then getting smacked down. But then a different cell will start making viruses and then get smacked down. And then a different cell will start making viruses and get smacked down. So herpes and HIV are kind of known for this. That's why people have herpes outbreaks, because the virus never quite fully disappears because your immune system can't catch all the cells with herpes inside of it. Likewise, same deal with HIV and why it's really hard to remove permanently from the body. So HIV is kind of one of the most well-known viruses when it comes to evading your immune system. And by understanding it, you kind of understand the immune system better. And why it's so successful is the fact that it attacks your helper T cells. And because your helper T cells that are so critical for helping to engage your immune system are destroyed, your cellular response and your humoral response drops way down, and then your immune system activity reduces quite a lot. And now you have a weak immune system. And generally speaking, The disease that comes, AIDS, from an HIV infection is a result of having a very weak immune system, reduced, almost absent T cells, and then to the point where people kind of start dying from AIDS. It's usually they're dying not of AIDS itself, but of infections that normally your body would shrug off. They can't because their immune system is trash. So just put it in context. Now, a heads up for COVID, and we're talking about cellular immunity right now. You might hear a lot about antibodies, especially when we talk about the vaccine, and they're like, the vaccine doesn't produce antibodies or enough antibodies, or the new variant reduces the amount of antibodies produced, or the antibodies suddenly disappear over time. And especially in the early fall and summer, people were freaking out a lot about COVID and how quickly the antibodies dropped off. But what we often forget is cellular immunity, T cells and B cells. Oftentimes people talk about antibodies as though it's the only thing in your immune system and your specific immunity, but it's not. The reason why we usually talk about antibodies is because it's way easier to measure. It's way easier to measure. It's a proxy for measuring how well your immune system responded to something. Because generally speaking, if you've made a lot of antibodies, you've also made a lot of B cells. And usually, because you made a lot of B cells, you also happen to make a lot of T cells, like helper T cells, to activate your B cells. So people use antibodies as a proxy because antibodies are really easy to measure in your blood, while B cells and T cells are not. However, then what ends up happening is people freak out over antibodies numbers, where in fact, it actually looking more and more that when it comes to COVID-19, your cellular immunity is actually far more important and vastly superior when it comes to reducing the damage of COVID-19 in your body and at least preventing harmful COVID-19 in your body. Antibodies are really critical for neutralizing and preventing reinfection, but once your cells are infected, having lots of cellular immunity is going to prevent that COVID-19 virus, SARS-CoV-2, from actually developing into a disease or at least into a serious illness. And vaccines, while they do also help your antibodies, they actually, all of them are, I would say, I've looked into almost all of them, especially the RNA vaccines and the DNA vaccines and the protein one. All of them have a method for engaging and stimulating your cellular response. Okay? So even if you hear a lot of stories about the antibody numbers, please remember that it is not exclusively antibodies that are critical to your immune system, but it's a cellular mediated immunity. And as long as the vaccines or COVID infection helps create some kind of cellular immunity, you're going to have at least a better secondary or even tertiary immune response to helping reduce the impact of COVID-19 if the virus is introduced into your body. Okay? But just remember, the first time you engage with an antigen, you create a decent amount of antibodies, but it does drop off without re-engagement. And because COVID-19 is still kind of unpredictable, this is why even if you have been or someone you know has been infected with COVID-19, it's still encouraged that if they qualify to get the vaccine, they should get the vaccine to produce that secondary, very strong response that prevents their body from getting reinfected in the future, especially if they get older. All right, so that concludes the lesson. I hope you learned at least a little bit about cellular immunity and how it relates to what's going on right now. Bye.